Good morning, and welcome to Symposium 1 of the Brain Initiative Investigators Meeting. I am Grace Hwang, a senior neuroscientist at the Johns Hopkins Applied Physics Laboratory. I want to first thank the NIH and the organizing committee for all of their hard work making this virtual meeting a reality. I'm delighted to be moderating a discussion of how we might achieve reciprocal advances in dynamical systems neuroscience and machine learning. Over the first hour, you'll be hearing brief talks from 6 researchers: Conrad Cording, Joseph Monaco, Kanika Rajan, Zach Pitko, Brad Pfeiffer, and Nathaniel Daw. The talks will be followed by a live Q&A session with our speakers. At any time during the pre-recorded talks, you are encouraged to type questions in the Q&A box and specify which presenter your questions are directed to. We will answer audience questions in addition to the general discussion during Q&A. Now, theoretical neuroscience and artificial intelligence have long inspired each other. An important common ancestor was the McCulloch and Pitts binary neuron model of logical computation in the 1940s, which was combined with Hebb's exposition of associative synaptic modification to inspire Rosenblatt's multilayer perceptron in the late 50s and 60s. This work set the stage for neural approaches to AI, but neuroscience and AI diverged in the ensuing decades. The past decade has seen surprising progress in AI that began with the advent of GPU computing technology and the availability of massive labeled data sets and deep convolutional networks inspired by the feed-forward architecture of the ventral visual stream. In fact, architectures and training schemes such as transformers and self-play have produced world-changing feats in language learning and game playing. However, this machine learning paradigm of AI, though impressive, is underpinned by continuous incremental improvements of architectures, optimizers, and objective functions. Thus, it remains unclear whether this is the road to human-level features of intelligence, including sampling efficiency, abstraction, and generalization. So today we ask our panel, can dynamical neuroscience, the study of temporal change and functional interactions across multiple scales of neuroactivity, help us bridge this gap between today's AI and the flexible biological intelligence demonstrated in the brain? We have convened an excellent panel of theorists and experimentalists with diverse backgrounds in computer science, cognitive neuroscience, neurobiology, physics, biophysics, and neuroengineering to share their perspectives on reciprocal advances in neuroscience and AI research. Now, I'm delighted to introduce our first presenter of our symposium, Professor Conrad Cording of the University of Pennsylvania. He will discuss how current dynamical systems views may be misleading. Welcome, Conrad.
As today's meeting is about how the dynamical systems neuroscience views can inform machine learning, I thought it would be great to kick this off by talking about the potential problems that the field still has to struggle with and that the field is addressing at the moment. So let me first summarize kind of the setup. So the brain is undeniably a dynamical system. Now we have 10 to the 10-ish neurons. They all interact, each of them with maybe thousands of neurons on average, but we cannot observe that overall system. In fact, we can only record from a relatively small subset of dimensions. And also, if we record individual neurons, it looks like they're very noisy. Now they spike and there's Poisson noise on that and so on and so forth. And hence what we usually do is we do dimensionality reduction, and in this lower dimensional space, we can now meaningfully analyze the dynamics of the brain. So what has just happened? There's a scale transition. We have a small scale where the dynamics happen and we have strong intuitions for that. And we just did a scale transition where we went from this microscopic scale to a mesoscopic scale where we instead look at the relatively small number of components. We do not currently really understand what we lose in that transition. Like it's clear that there's information lost as we go across those scales, but which of the questions that are meaningful at the microscopic level are now still meaningful, and which ones are not. It's a central question to the field, the scale transition. Some people argue that the scale transition cannot meaningfully be done, and it's a central issue that we have to grapple with, and it would be nice to see more work in that direction. So first, let's talk about this setting. In general, in the field, it is agreed that neural representations are low dimensional, and we see the literature full of graphs like that. How do we traditionally describe that? Well, we do linear dimensionality reduction, singular value decomposition. So what we have here in the graph is on the y-axis, we have the variance explained by a singular value, and on the x-axis, we have the index of that variable. And what we find is these graphs usually look what we see here. We have a few really big singular values, and then we have like this tail that hangs over it. And we usually use some statistical criterion to say, well, these are significant and the others are not. But of course, if this is the domain in which we are, what would happen if we had more data? Well, we would be able to get more components significant. So in that sense, this kind of analysis that is very popular in the literature does not ask what's the dimensionality of the neural activity. It just asks, can we give a lower bound on the dimensionality of the data? But that very much depends on how much data we get. And in fact, empirically, as we get more and more data from the brain, we find that we find more and more of them significant. And in fact, here I want to just show some really cool new analysis by Carsten Springer and a good number of collaborators, where instead of the usual approach of recording a small number of relatively noisily signals, they recorded lots of signals at relatively high data quality. Now, on the x-axis of the left plot here, we see the dimension of how many axes of the principal components. So basically, the items to the left are the early principal components, the right ones are the high principal components. So what we find isn't that there exists some significant principal components and some non-significant ones. No, we find like a broad power law with the coefficient very close to 1. What that means is that there's no sign of strong low dimensionality here. And in fact, in that same paper, we see good reasons why this is so. So in typical studies, we only give a very small number of stimuli, typical maybe 32 images or maybe movements of the hand into 4 different directions. And the principal components, if we use this impoverished lab type situations, we only need a very small number of them to capture the vast majority of the variance. But as soon as we go towards the real world, it looks like we need far more dimensions. Now, caveat, of course, we cannot be sure about that. And let me highlight why. So we need more research on the question of what's the dimensionality. It's central to our field. Not like how far we can push various approaches in dynamical neuroscience very much depends on how low dimensional, high dimensional the data is. It's unclear at the moment how low dimensional it is. Most studies that are being cited to argue that it is low dimensional do not show it. For example, there's a lot of decoding studies that say drive a brain-machine interface. They often find that if we only take the first few principal components, it does better than if we use all of them. Well, the early ones have, by definition, high signal to noise ratio. So it's kind of not a meaningful comparison because it muddles the dimensionality of the signal to the properties of noise. And it would be really, and also it could be that the brain is very low dimensional, it just unfolds in a high dimensional space, and there is a curved manifold in that space. But it's very hard to know the analysis there. So I think that we cannot be sure at the moment how high or low dimensional the brain is. Next thing is, even if the brain is relatively low dimensional, it's not sure if the dimensions that we can pull out really tell us the interesting dimensions. So what we did here is we took a simulation of the MOS 6502, and I should mention that is Eric Jonas who did that. And we can record from all the transistors in that microprocessor, and we understand pretty well how the microprocessor works. works. So what do we find? Well, we do find reflections of the clock signals. We do find reflections of, say, the fetch and the read-write signal in it. But that whole fine-grained dynamic of how computation actually happens on the microprocessor, we cannot meaningfully make sense by looking at such data. Now, that doesn't mean that it's not possible. It just means that with the techniques that we have at the moment, it's not possible. So does looking at dimensionality reduction pull out the dimensions that are really relevant to understand the system? We don't currently know that, and we are lacking theories that tell us why we should believe that. There's also the problem of confounding. So this is something that occurs at many layers. It just as well exists for dynamical systems ideas. So let's take the model. When Conrad drinks beer, it lowers the voltage of the transistors in his computer. Well, there's supporting data. Turns out that when Conrad drinks beer, the voltage is on average lower than when Conrad doesn't. So we could come up with models which say Conrad's beer drinking lowers the voltage of that. But the problem there's of course a confounder, which is the time of the day affects what Conrad does, and the time of the day also affects if he turns his computer on or off. And therefore, this confounder describes both. And therefore, any causal relationship I would have described between two such variables can be meaningless. The same thing is true for the brain. It could be that we see relationship between variables. We know by definition, because we only record a small number of dimensions of the brain, that we miss dimensions, that we don't know about a lot of dimensions. It can very well be that these dimensions make it happen and that we see the reflection in our low dimensional projections. Now, even if there wasn't a problem, so let me summarize now the neurons view and the manifolds view. So if we describe the brain in terms of neurons, yes, we have far more, but the interactions between neurons are sparse, and we kind of understand the nature of that sparseness. We know how many synapses a typical neuron does. Once we take all those neurons and we dimension out to reduce them, then the connectivity pattern that was sparse in original neuron space is very likely to be dense in the manifold space. On the other hand, manifolds are less noisy, as I mentioned before, than individual neurons. Now, individual neurons can be feasibly perturbed, whereas with manifolds, that's almost impossible. What does that even mean to perturb a manifold? Now, in individual neurons, we might be able to understand the nonlinearity based on models of, say, dendritic processing. When it comes to manifolds, it's very unclear how we should think about what would be the right nonlinearity for them. And with neurons, we can feasibly count them. For manifolds, it's very hard to know how many we have them. And I'd argue that we have very good intuitions for arguing about neurons and not so good intuitions for arguing over manifolds. And now, here's another problem. So even if there were low D and we had intuition, causality is a major problem. If we want to build a machine learning system that somehow mimics the brain or is inspired by the brain, we want to know how information flows. At the moment, there are no tools apart from perturbations that reveal the flow of information in such networks. As we usually aggregate data across neurons, we lose the small number of tools that we currently have to get at causality, for example, looking at the precise timing between neurons. What I'd argue is that we might instead want to focus on cost functions, learning rules, and architectures, but that's a different talk. Look at the Richards et al. Paper. Now, I want to skip over this, I apologize. Now, the question is, what are the needs of the machine learning field when it comes to neuroscience? What we miss in machine learning is we lack good ways of handling causality. And we should focus on how the nervous system is so good at understanding the world in terms of cause and effect. We lack good ways of lifelong learning. And how can we keep getting better? Why do people not catastrophically forget? We lack good ways of active learning. You know, like babies know what's interesting and what's good for them. We're really good at like trying to soak up the information that's good for us. Current machine learning systems are very limited at that. And as we focus on dynamical systems neuroscience, it's important for us to focus on the research that gets us towards these questions. And after giving all these hard problems that this talk was about, I'm very much looking forward to the rest of the symposium. And now after this talk, it's with great pleasure that I announce the talk of Joe Monaco from Johns Hopkins University, who asked, can transitory neurodynamics verify learning theories for brains and machines? Very much looking forward to his talk.
Thanks for kicking things off, Conrad. I'd like to thank Grace Hwang and the conference organizers for inviting me. I'm a theorist and computational neuroscientist. In this talk, I'd like to highlight several gaps in our dynamical understanding of the brain to address the question, can transitory neurodynamics unify learning theories for brains and machines? To start, I'd like to show a visual contrast between learning processes and artificial and biological neural networks. On the left is a reduced visualization of a standard convolutional network being trained with backprop on the benchmark MNIST dataset. On the right, a beautiful two-photon video of mouse CA1 hippocampus is shown as the mouse learns an olfactory task. The dense activation across the layers of the artificial network stands in clear contrast to the sparse activity in the hippocampal circuit. There are many more major differences that could be listed, which is what I've done here. So the details could be argued over, and this is certainly not exhaustive, but there are many potentially interesting gaps in which neuroscience might be translated to advances in AI. I've highlighted three in particular that I'll focus on here. The first gap, related to timing, points out the prevalence of local oscillations and phenomena such as task-dependent modulation of phase coherence. In particular, oscillatory coding by spike timing or phase coding has been extensively studied in the hippocampus as the phenomenon of theta phase precession. Brad Pfeiffer's talk later in the symposium will address more on that. The second gap relates to observations and theories that recurrent connectivity in structured networks can give rise to collective emergent behaviors, which are both self-organizing and self-stabilizing. The dynamical notion here is that memory retrieval is a state space trajectory that searches and probes the energy landscape of memory networks. The third gap is a deeper question related to information versus meaning, for which I will speculate that volitional interaction with the world is what allows animals and biological brains to construct meaning. Two well-studied ideas from neuroscience are suggestive. First, motor efference copy may be interpreted as sampling a forward model to predict the sensory consequences of actions. Second, the sensory reafference principle implies that those forward predictions may help separate external sensation from sensory changes due to self-driven actions. So this separation is critical to perception and speculatively to the sense of agency. A tremendous amount of brain structure is generally devoted to these motor sensory feedback loops. As further speculation, I would consider that these reafferent predictions might be implemented as dynamical, high-dimensional state transits among low-dimensional attractors in distributed memory networks. Those would be networks with conjunctive action perception encodings that have been acquired through experience. So one question that I've asked related to timing is, how do landmarks reset path integration during navigation? And could resetting be supported by oscillatory coding outside of the hippocampus? This project was a collaboration with Tad Blair at UCLA, who collected the data, and Kechen Zhang at Johns Hopkins. Path integration computes spatial position relative to some previous reference point. Thus, it naturally accumulates errors. So the internal representation for this journey shown here on the left may look like this green line, where the trajectory drifts off course over time and needs to be corrected at the next landmark. It's been unclear where path integration is exactly computed or how it is reset. So Tad recorded theta-rhythmic neurons from the subcortical areas in the lower two rows of this diagram. And I focused on his data from lateral septum here, which is the major subcortical output target of the hippocampus. So interestingly, I found cells like this with clear spatial modulation of spiking, where spikes are shown as the red dots on top of a trajectory. By averaging the activity at each location in space, we can create maps. So we can see that the standard firing rate map up top shows the rate code of this cell, but the map on the bottom shows the mean theta phase of spiking with a clear correspondence between the two. I quantified this with rate phase correlations, revealing that higher firing rates were strongly and monotonically associated with earlier firing within the theta cycle. So I called cells like this phasor cells. This is distinct from hippocampal phase precession, which has a highly asymmetric rate phase relationship across the place field. This symmetric coupling implied an intuitive phase coding mechanism, which I implemented in dynamical bursting models, combining theta rhythmic inhibition with external inputs, as in the purple voltage trace here, shown for both low input in the top plot there and high input on the bottom. So sampling different inputs recapitulated that phasor cell correlation. Then, to create a large network layer of model phasor cells for the open arena, I trained a generative data-driven model and synthesized 1,000 phasor cell responses, and some of which are shown here in these rate map phase map pairs. I did this in order to address the question of learning. So this bursting target neuron, shown here in the top left, is a bit noisy, so its theta phase randomly drifts around. As a teaching signal, I created 64 sinusoidal gratings corresponding to path integration with differing spatial offsets. So training target neurons to these patterns resulted in these responses, where each video frame that you can see is the phase map of one of these target neurons. To perform resetting, these responses must support reconstructing the animal's trajectory. Using Bayesian decoding of burst phase, the sample trajectory in blue was poorly reconstructed, only along the single preferred direction. Instead, when trained against all different preferred directions, reconstruction was possible with much higher fidelity. Thus, I theorized that an alternative subcortical pathway for resetting could operate using mechanisms of collective synchrony. In discussing these results with colleagues at Johns Hopkins Applied Physics Laboratory, one of them, who is the moderator of our symposium today, Grace Hwang, thought the phasor cell mechanism could be translated to the domain of robotic control. We received an NSF Brain Initiative Award to study this possibility in the context of robotic swarms. There are two key points to understanding our swarming framework. First, we analogized individual agents to single neurons, such as phasor cells, and groups of agents to neural circuits. Second, relative motion was directly realized as a form of Hebbian learning, using spatial kernels to convert pairwise distances between agents into synaptic weights and vice versa. The neural mechanisms consist of a feedback loop between a neural activation variable P and a phase state theta. So agents integrate input from cues, which are the purple shapes in the map shown at the lower right here, and from rewards, which are the gold stars in that map. Recurrent inputs from other visible agents are phase coupled such that similar phases mutually excite the agents and opposing phases suppress them. Thus, each agent is a simple model of rate phase coupling, similar in principle to phasor cells. To drive motion, we implemented Oya's rule for the recurrent swarming matrix shown at the top here, and a feed-forward matrix allowing approach to rewards, those gold stars, on the bottom. So each of these two learning rules has a relevant presynaptic variable or presynaptic input, and the activation provides a postsynaptic output. So these are the two factors that we need, the correlation that we need to drive Hebbian-like learning via Oya's rule. Now, we can simply initiate 300 agents at different positions in the environment, and they're shown as circles colored according to phase. So here's a simulation video in which you can see that the initial clusters rapidly expand outwards to form lines and arcs. So the agents are phase sorted within these structures, and these structures self-organize in complex patterns over time. Note that every spatial configuration of the agents corresponds to a new attractor map. Often, several agents will mutually synchronize to the same phase and location, forming a local fixed point, which can then destabilize due to nearby agents. In this simulation, only one gold star reward is active at a time. So this is a dynamically changing environment. The resulting responsiveness of the swarm is akin to dynamic replanning, which is a critical topic in autonomous control. No offline pre-training is necessary. This motion is an online dynamical learning process. Thus, swarm motion can be expressed as Hebbian learning in an oscillatory attractor map network. With a small modification, we can operate a single agent, the larger green circle seen here on the left side of the map. This agent is guided by a swarm of virtual particles constituting the agent's internal mental model. This paradigm completes the neuro to AI loop by bringing the neurally inspired artificial system back into the standard animal in a maze modeling paradigm of theoretical neuroscience. This approach may benefit neuroscience by visualizing neural circuit dynamics in a completely new setting in a way that improves our theoretical intuition and creativity. I hope this illustrates at least one way to reciprocally advance learning theories in brains and machines. And with that, I'm really looking forward to the rest of the symposium. The next speaker is Kanika Rajan from the Icahn School of Medicine at Mount Sinai, who will be speaking about recurrent neural network models of adaptive and maladaptive state transitions.
Hi, I would like to begin by thanking the organizers of this wonderful panel. Thank you for the opportunity. The title of my talk is Multi-Region Models of Adaptive and Maladaptive State Transitions. And so a big thrust in my lab is to look at data sets like this collected across different nervous systems and ask the following question, which is, what are the circuit mechanisms that are conserved when you go from a small nervous system such as a larval zebrafish to a larger nervous system such as the rodent, even non-human primate or monkeys, and which of them are which of them are different. So both ends of these questions are deeply interesting to me as a theorist and my colleagues in my lab, but for the purposes of today's talk, I would like to narrow this down to this one question, namely, what circuit changes mediate the state transition from active coping to passive coping in the larval zebrafish? So to get at this, my approach is to first build and then analyze network models that directly use these time series measurements that are obtained experimentally as constraints. So the models are built by using the constraints of the experiment directly. And then once I build these models, I'm able to get at circuit changes or mechanisms from reverse engineering these models that I can't get from measurements alone, or the experimental colleagues can't just discern from measurements alone. And so that's what I'm going to tell you about today. So the first thing I do is to build multi-region network models that are able to capture the interactions both within individual regions as well as long-range brain-wide communication principles or inter-area interactions. And so here's a very simple schematic of what a single region like that would look like. It's a recurrent network model where each unit is a model neuron. They're all connected all to all through recurrent connections. The connections between all the cell pairs are expressed in terms of a directed interaction matrix, which captures the pairwise strength of the connection between or the interaction between individual units in this network. Now I can extend this to two region models in which I can take two recurrent network models built exactly like I explained before and wire them together reciprocally. And the directed interaction matrix has a slightly more complex structure in which the within region connections are along the diagonal of this matrix, and across region interactions are the interactions that are in the blocks that are on the off diagonal elements. Now I can play this game potentially brain-wide, and I can build a multi-region RNN that is as big as the whole brain of, in this case, the larval zebrafish. And here's an example showing 4 of those, of what that would look like if I looked at just 4 of those modules connected reciprocally, and what the directed interaction matrix within such a network would look like. So that's the first thing to do. The second thing to do is to take the activity of each unit in this artificial model network, which is initially irregular, as you see in the red trace on the right, and then train it to match a target that's derived directly from the experiment. So in models that I build, the network is usually as big as the size of the experimental data set, which is what you see as the N of the whole brain, as you see here. In this case, it's the order 10 to the 4th number of units. And initially what starts off as irregular activity from these artificial networks is trained by using recursive least squares to match the calcium activity directly obtained experimentally. So it's a directly data constrained network architecture. And through the process of learning, the network connectivity changes through this delta Jij term that you see here. So what does this exercise buy you, right? We've built a model that actually has the same level of complexity as the experimental data set in terms of number of neurons, and the time series that it produces matches that of the experimental data set. What it buys you is 3 different things. And before I tell you what they are, I want to highlight a few papers that my lab has written very recently in terms of building networks that are constrained by neural dynamics, as you see here, as well as recurrent networks that are constrained by behavioral data as well. So we've done quite a lot of work in this, and we're really excited to push this direction forward. So what are the things that such an activity buys you, right? It buys you three things. One is you build a model that produces realistic neural dynamics. So once it's trained, it's autonomously producing the dynamics that are consistent with the experiment. You're not actually fitting a stationary curve to anything. You're producing the entire dynamics autonomously. The second thing is that you can look under the hood of this trained network and you're able to infer directed interactions through the matrices that I showed you that are consistent with producing the dynamics that were observed experimentally. So things like effective or functional connectivity, except while it captures the entire experiential state. And the third thing, which is what I'm going to focus on today, is a new direction that we're pushing, which is that this gives you the ability to look at currents due to the recurrence within and between regions. And so this is something, and the two things that I just mentioned before, the connections brain-wide as well as the currents due to recurrence are things that you can't obtain through measurements alone. And so what do the currents look like? And so here I'm showing you a very specific example. I'm showing you a 3-region model that we just built, one with the habenula, one with the raphe, and one with the telencephalon. Those are the 3 regions in this network model. And a model like that gives you, as I said, those 2 things kind of trivially. The first is the activity that is similar to data, because of course that's what you're training the network to do. The second thing you get is this directed interaction matrix. In this case, it contains 3 blocks along the principal diagonal. The red block in this cartoon is schematized by, represents the within habenula connections, the green block, the raphe connections, and the blue block, the telencephalon connections, and the between regions are in the off-diagonal elements. Now, the dot product of these 2 objects gives you the third thing, or the currents due to recurrent interactions. Now, what these currents can do, if you look at the term carefully, is by looking at just different summations, right, along the index J, you can look at currents both within a same region or currents arriving into this region from different areas. So, for example, if I'm considering just the habenula, by summing this only over the habenula neurons, I can look at the currents that a habenula neuron receives from other habenula neurons within the habenula, or I can look at the currents that the habenula receives from the raphe, or I can look at currents that the habenula receives from the telencephalon. And so I can sort of separate the overall activity obtained experimentally into its different components. And that's what I'm schematizing here. And what do they look like? Right? So the habenula to habenula currents in this 3-region model that's been fit to a shocked fish in this case look like this. So I'm plotting 2200 habenula neurons as a function of time. And here, just for ease of visualization, they're sorted by time of activation. The orange dots on top symbolize the time periods at which the shocks were presented to this particular fish. The raphe to habenula currents look like this. Here, the units are actually sorted in exactly the same order as the first one, and the telencephalon to the habenula currents look like this. They're again sorted in the same order as the very first of these graphs. Collectively, if you sum all of these, they should look like the overall currents experienced by the habenula. So there's a vast amount of literature in doing dimensionality reduction on experimental measurements or calcium activity or spiking activity or electrophysiology of different kinds. What we're going to do now is use the exact same techniques, except now we're going to apply them on these source-separated currents and derive these 3 axes. So all we've done is do principal components on these 3-component currents and derive those axes. Now we can take the activity recorded experimentally and project them onto this new coordinate system. And that's what that looks like. So now I've projected, so that's what the black trajectory is the activity, is the overall activity of this particular shocked fish. And on top are different dots that are colored by whether they arrive early in the experiment or they arrive late in the experiment. And something surprising happens here. As soon as we plot the data like this, it looks as though the early shocks are mostly, the early shocks influence the trajectory in the raphe to habenula subspace. And it's only later on in the experiment or the experiential state that the habenula to habenula subspace and the telencephalon to habenula subspace get populated. So what we're able to do is to look at things like inter-area effects such as timing. So what I'm suggesting here is that the early part or the active coping phase of the behavior, as you see here, I've brought back the behavior diagram, where the control fish whips its tail vigorously to avoid the first shock, that seems to be influenced mostly by currents flowing from the raphe into the habenula, and the later part of the passive coping is driven mostly by the habenula to habenula and telencephalon to habenula occupation of the subspaces. So we're currently working on several manuscripts that are related to this, one on the methods and one on applying that to the fish data. But in our view, looking at source demixed input currents that reveal these interesting inter-area effects is much more informative than looking at the raw activity of the habenula neurons, the average activity of these neurons, or the much more traditional way of just projecting the activity onto the principal components, which is the much more traditional view. Now, if I look at the same exact currents in terms of time, what you see here is again a stark difference between the control fish and the shocked fish, which is what you see here now. The gray is just smooth movement, or the activity, or the tail whips that have been just smoothed for ease of view. And if you look at the shocked fish with me for a second, you see that at some point the movements have ceased. Now, in comparison with the control fish, the trend that we looked at in the overall 3D plot seems to be that the active coping, or the increase in activity of the movement, seems to be driven much more by the raphe to habenula engagement, or the currents, as you see in the average ramping in the green. And it's only later on, or the passive coping, that seems to be influenced by the habenula to habenula and the telencephalon to habenula currents. And there's no such a clear separation between the three in the control fish, in which the movements continue pretty much throughout the experiential state. And so with that, I would like to once again thank my colleagues and collaborators, particularly Matt, Camille, and Jean on the theory side, and Tyler and Aaron from the Carl Deiseroth lab from the experimental side. I would like to thank the Brain Initiative for their R01 so early in the formation of the Rajan lab. And without their help and support and their faith, none of this would have been possible. So go Brain Initiative. Before I wrap up, I would like to dedicate all my work to my parents, whom I lost very recently. They were my biggest supporters and the biggest allies to our scientific and all my work. With that, I would like to say one more thing, which is that if anybody is interested in work of this nature, in understanding how mechanisms arise by building network models that are constrained by data and reverse engineering them, then please join us. We're hiring aggressively. With that, I will leave it there. I want to introduce the next speaker, who is Zach Pitko from Baylor College of Medicine. And he's going to be telling us about algorithmic dynamics in population codes. So take it away, Zach.
Hi, everyone. I'm going to talk about algorithmic dynamics in population codes. The central claim here is that when trying to understand the brain, we can best interpret and generalize its computational dynamics if we use task-relevant coordinates within the neural populations. What you see here is a little cartoon of a neural manifold, and the activity of the neurons are proceeding through this wiggly black line. And we decompose those dynamics into two different sets of coordinates, one relevant for a task and the other irrelevant. So the irrelevant ones are going up and down in this picture, tracing out a ribbon as the task relevant coordinates follow along this S curve here. Which particular value you take in the vertical direction, the task irrelevant direction, doesn't matter. And so we can imagine doing a targeted dimensionality reduction where we squish that ribbon down to just the curve. And those are the computational dynamics that we need to characterize. Why is this the best description? Well, it's because of equivalence classes of computations. From the representational point of view, we have many neurons that are tuned to many variables. That is, it's a distributed code. And those neurons are redundant, so they carry the same information. And it doesn't matter which particular ones you pick, whether you pick these neurons that carry that information or some other neurons or some average of the two. So we can compress that information down and remove the redundancy by looking at the encoded representation with respect to the task. Now, transformation-wise, we know that neural networks can approximate any function given enough resources. That's universality. And conversely, any function could be computable in many different ways by a neural network. That's degeneracy. So do we precisely care which way it's actually computed? I think computationally we don't, and it gives us greater understanding if we take the more abstract point of view. Here's an example of this. Let's say we wanted to compute a quadratic function. Now that nonlinearity could be implemented through literal quadratic nonlinearities on neurons, and you add them together and you get the answer you want. Or you could use a bunch of piecewise linear functions with thresholds that are uniformly distributed. And if you add those together, then you get that same quadratic function, at least a close piecewise approximation to it. Now, what structure should we be looking for, and what variables do we care about? There's a hypothesis that the brain is matched to the world, that through evolution and development and experience, it has come up with a model of the world that's useful for its actions. And here we follow that hypothesis, and we say that the brain approximates probabilistic inference on a generative model of the world. And here we'll add a little bit of new stuff, using nonlinear message passing in redundant neuronal populations. Okay, so I'm going to talk more about what this nonlinear message passing is. First of all, when you're accounting for all the structure in the world or all the possibilities that you might need to do inference over, not anything is possible. And so the brain should and does use structure. One natural framework for thinking about that is probabilistic graphical models, which we can use to simplify a joint distribution over many variables given some observations O, with this S being the state of the world. And we simplify that by looking at interactions between those variables, statistical interactions. This allows us to simplify the whole distribution to smaller pieces with using this conditional independence relationships. Now, once we have that representation, that's much more compact than an arbitrary distribution, but it's still hard to figure out what you need to do with the variables of interest. And that's because not everything matters. So you have to marginalize. You have to essentially ignore the variables that are not relevant for the task at hand. And accounting for all of the variability of other things that might have influenced the task variable, that's what's hard. So this is something we need some kind of algorithm for. And this is where message passing inference is actually very valuable. This is an approximate inference. And what it does is takes the local evidence, the observations that you get, and summarize that for each variable and figure out how that variable is going to affect the neighbors. Send that information along the graph of directly interacting variables, and then iteratively update that process with new information as it comes in or as it spreads through the graph. The consequence of this is that message passing will localize information at the variables that are relevant so that they are actionable. You probably already know some message passing algorithms. This general category of algorithms includes mean field, belief propagation, expectation propagation. Even Gibbs sampling is a stochastic message passing function algorithm. And each message passing function is defined by its own nonlinear function updates. So what is the brain's nonlinear function? How can we characterize that? What we need to do is, from neural data and its input, to recover how the variables are encoded, that is the representation, which variables interact and how. That describes the implicit graphical model that the brain creates about the task or tasks. And crucially, how those interactions are used. That is, what is the message passing algorithm that defines how information flows through the graph according to the strengths of the interactions there. How is this remotely possible? I mean, I've just described a general purpose implicit nonlinear dynamical system. How is that not dramatically under-constrained? Well, it's constrained appropriately and identifiable because we have assumed a global algorithmic symmetry in asserting that there exists a message passing algorithm. And I would say that this is actually essential for any kind of understanding of an algorithm in the brain. What does it mean to understand something? It means that there is some algorithm which is shared across many different aspects of brain computations. And so we're sort of formalizing this mathematically. in a way that makes it identifiable to say, what is the algorithm? Here's a cartoon of how that would work, how you get this implicit message functions. We have some neural activity, which are going through whatever dynamics they do, and some special patterns represent different task variables in the world. Here is a variable, a green variable. And then other patterns will have their own representations, their own encoding of something else that they're representing. And as information flows through the neural dynamics over here, that corresponds to information flowing on this implicit graphical model, which allows probability distributions to interact with each other, probabilities about subsets of variables at a time. These correspond. So an example application of this would be, let's say you're looking at something which is difficult to discern, and you have a couple of edges in an image which are described by some orientation. You can get more information about what's missing between them, or you can fill in the blanks, or you can resolve some of the ambiguity by allowing information to flow across space according to this graphical model. I show here a couple of scatter plots illustrating that for a simulated brain, we're actually able to do this. We have a completely distributed nonlinear dynamical system that implements some inference algorithm. And we follow some expectation maximization algorithm. We're able to recover both a good description of the neural dynamics and a good description of the latent variables. Shown in blue, you get this diagonal, the inferred and the true match. That's where you get the diagonal. So to conclude, we have this, this way of thinking about computational dynamics in neuroscience that might be informative about machine learning, because we can learn what kind of tricks the brain uses in its implicit inferences operating on its model of the world. So at the computational level, this is approximate inference that uses it's a mathematical framework for what agents might compute while they're trying to perform tasks. And at the algorithmic level, we have this recoding dynamics where the code itself is changing. What the task variables represent is changing as they pass information around and refine their estimates of the world. And this is because latent variables are represented across neurons, and the nonlinear dynamics performs implicit inference. So this is a little overview of some work that you can read about in some other sources if you look up my name. And this is the contributions have been coming from a bunch of people in my lab, including Raju, Ki-Jung Yoon, Qianli Yang, and Zhe Li, and collaborators Scott Linderman and Dora Angelaki, as well as several other people who have been influential in thinking about these problems. Thanks to the funding, and thanks for your attention. And next up, we have Brad Pfeiffer from UT Southwest, who is going to tell us about hippocampal replay dynamics.
Hi, I'm Brad Pfeiffer. I want to start off by thanking Grace and the other organizers for putting together this meeting and for giving me the opportunity to talk today. The people that were involved in this project are Hwang. She's a senior graduate student in my lab who performed many of the analyses for this project. And the data that were used in this project were generated by myself when I was a postdoc with David Foster, and he has very graciously allowed me to take these data with me into my own lab. So imagine a sequence of reciprocally connected neurons shown here. And imagine that this population of neurons is repeatedly and reliably activated in the following pattern, A, B, C, D, E, F, G, H. What we understand about spike timing-dependent plasticity is that if presynaptic neural activity precedes postsynaptic neural output, you should potentiate that synaptic connection. So in this example, you should potentiate the forwards sequence that was just expressed. This is obviously very useful for prospective evaluation, so that at some point in the future, if neuron A fires an action potential, through a syn-fired chain-like activity pattern, you might reactivate neuron H, which would allow the neural network to make a prediction about what is upcoming. However, spike time-independent plasticity has a depression component as well, so that if postsynaptic neural activity precedes presynaptic neural output, what you should do is weaken that synaptic connection. So this makes it very challenging to imagine how the hippocampal circuit might retrospectively evaluate the causes of some current reward or negative salience. And yet we know that during periods of rest, during sharp wave ripples, the hippocampal circuit is very good at generating reverse ordered sequences in a phenomenon called reverse replay. And although many models have been put forth trying to explain how one might achieve reverse replay, I think it's safe to say that we don't fully understand how a neural network can generate reverse ordered sequences following forwards ordered activity patterns. And this is what our project will be focused on. All right. So theta sequences are an ongoing oscillation within the hippocampus and other brain regions that are about 10 hertz. These organize patterns in the hippocampus, at least, of place cells into specific sequences, as shown here on the bottom. The y-axis is position on a linear track across time. The black line is the rat's actual physical position as it runs, in this case, from the bottom to the top of this linear track. And the color code is the result of a probabilistic decoding algorithm. So this is the posterior probability, basically the location of highest representation in the brain at any point in time. And hopefully you can appreciate that these theta oscillations are organizing hippocampal sequences into these sweeps of spatial information that progress ahead of the rat on virtually every theta oscillation, allowing for perspective representation. And these sequences seem ideal for establishing synaptic plasticity across a neural network in the forwards direction. But labs such as Laura Colgan and David Reddish have suggested that theta sequences may also encode some kind of retrospective encoding. And we wanted to see if we could identify some features of retrospective representation in our own data. So we decoded data from several rats where we had recorded from several hundred simultaneous place cells. And here is a result of the decoding for one small epoch of time. Here are 3 consecutive theta oscillations. And what I'm showing you is the result of the probabilistic decoding algorithm. This is the posterior probability. These have all been normalized according to the rat's physical location at the time of the decoding. So 0 is always the rat's current location. Positive values are ahead of the rat and negative values are behind the rat. And you can see these sweeps of activity representing prospective evaluation on every theta oscillation. But hopefully you can also appreciate there's this negative, this reverse component on each one of these theta oscillations. This is also true in the open field. It's a little bit more noisy because we're compressing a 2-dimensional environment down into a single dimension, but still positive values are ahead of the animal, negative values are behind the animal. And you can see this checkmark-like pattern where each theta oscillation has a reverse component followed by this larger forward sweep. Those are not rare events. We see these in every single rat in every behavioral paradigm on virtually every theta oscillation. And here's the average across all of the many tens of thousands of theta oscillations that we've analyzed. We've restricted our analysis only to times when the rat is running 10 centimeters per second or faster in either open field or linear track sessions. And hopefully you can see just by eye, there appear to be 2 distinct components to these theta oscillations if you decode with a sufficiently large number of place cells. We can identify maximum and minimum posterior probabilities across these windows of decoding. And this is basically a representation of the quality or the accuracy of the spatial decoding at any point in time. And we can use these minima to define analysis windows so that we're using the data to define these analysis windows rather than defining them arbitrarily. And we can then quantify various aspects of spatial representation within these analysis windows. And we can see that there's clearly a reverse window that encodes a backwards sweep of spatial information and this canonical forward window that has been well studied by other labs. Importantly, the reverse window moves backward faster than the rat is physically running forward. So it's not the case that the hippocampus is representing a static location that the rat is running through, but rather this is on every theta oscillation, the hippocampus is iteratively encoding, as actively encoding a backwards evaluation of what just happened, followed by a forwards prediction of what might be upcoming. We can, of course, quantify statistical significance in both the forward and the reverse window, and both of those paths, forward or reverse, are statistically unlikely to have arisen by chance. Next, we wanted to ask what might be the underlying mechanism for the encoding of this novel reverse component. We were struck by the fact that the reverse window of the theta oscillation comes at a phase of theta, the peak of theta, associated with minimal firing activity across the hippocampal network. So here's a firing rate across theta phase plot for a typical hippocampal place cell. You can see this neuron is relatively inactive during this reverse window. So we wondered if perhaps there was a subpopulation of hippocampal place cells that might be selectively active during that window. And that's, of course, what we found. Most cells that we looked at showed a unimotor relationship between firing rate and theta phase, and they were inactive during the reverse window. But we also saw a relatively large minority of cells that we called bimodal cells that fired twice in every single theta oscillation. And one of these peaks was specifically associated with the reverse window of the theta oscillation. By the way, bimodal cells had been shown previously in work from Yuri Bujaki's lab, and they identified that cells that reside in the deep portion of the pyramidal cell body layer typically fired twice in every theta oscillation. And we have other evidence to suggest that bimodal cells in our data are very likely the same deep pyramidal neurons. Across our data set, we see that about 1/3 of the neurons that we record, regardless of RAT and regardless of behavioral behavior type, are bimodal cells. I'm not going to show the data, but the backwards component of the theta oscillation is very heavily, almost exclusively driven by the activity of these bimodal cells. Bimodal cells are more likely to participate in replay than unimodal cells, and if you just look at activity of bimodal cells, they are more likely to participate in reverse replay than in forward replay, suggesting that the activity of these cells during that reverse theta oscillate, the reverse component of the theta oscillation, might establish synaptic plasticity, which allows for this sort of retrospective evaluation during offline periods. We then wanted to ask, what is the underlying cellular mechanism that allows for the expression of this reverse component? A lot of work has been done studying the forward component of theta oscillation, and it's believed that phase precession might underlie that forwards component, as shown in this diagram from a paper from Bruce McNaughton's lab. So phase precession is the phenomenon where as a rat runs through a place field, the cell fires earlier and earlier in consecutive theta oscillations, and at a population level that generates sort of for free these forward sequences. So we wondered if phase procession might underlie the reverse component, and that's exactly what we found. Unimodal cells shown here display a clear phase precession phenotype. So as the rat runs through the place field, these cells fire earlier and earlier in the theta phase. However, bimodal cells show two distinct clouds of activity as the rat runs through the place field, one that displays canonical phase precession and one that displays phase procession. We again defined windows of analysis based on minima in firing rates across theta phase, and we can quantify statistical significance, and we see that the phase procession component is indeed more positively sloped than you would expect by statistical chance. All right. Finally, in the last few seconds, let me just emphasize that the forward and the reverse windows appear to be statistically independent of one another. So if we rank each theta oscillation simply by the amount of firing in the reverse window or the forward window, we can then quantify various aspects of spatial representation. And I don't have time to go through these data, but basically, when we rank them by activity in the forwards window, the quality of the forwards window improves, but the quality of the reverse window does not change. Or if we do the opposite, if we rank them by the activity in the reverse window improves, but the quality of the forward window diminishes, suggesting that there's an independence, or in some cases, an anti-correlation between the quality or the activity in the forward and reverse window, suggesting that these two types of activity are driven by distinct circuit-level inputs. All right, so to very quickly summarize, we see theta sequences encode both forward and reverse sequences. The reverse component is driven largely by the activity of bimodal cells. Bimodal cells encode this reverse component via phase procession, and the forward and the reverse windows appear to be statistically independent of one another, suggesting that they are driven by distinct synaptic inputs. These are the people in my lab. These are my funding sources. Thank you very much for your attention. The next speaker is going to be Nathaniel Daw. He is the Hwang Professor in Computational and Theoretical Neuroscience at Princeton University, and his talk is entitled Balancing Flexibility and Reuse of Computation in Reinforcement Learning. Thanks.
Okay, today I'm going to give a very quick overview of some computational modeling I've been working on with Hwang Array. And it's available as a preprint here if you'd like to see the details. So I study how the brain solves trial and error choice problems, what computer scientists call reinforcement learning. So in general, the tasks we're interested in have multiple steps, like mazes, like this one, where you have to make a series of turns to find the goal. And both organisms and computers need to solve an objective function, like this one, that adds up rewards over multiple steps over different trajectories. And solving or learning or computing this function turns out to be sufficient for finding the best action. It says how much reward to expect for different possible long-run consequences of different actions. But this involves a lot of computation. And one issue that's gotten more attention in biology than engineering, I think, is that organisms need to sort of autonomously manage this computation to decide when it's worth doing and when to trade off against shortcuts that involve reusing previously computed actions, but may be inflexible and make mistakes. So for instance, we have computationally heavy so-called model-based strategies that literally solve this equation by iteratively enumerating different possible trajectories and adding up rewards, like sort of step-by-step mental simulation. In the spatial case, these are associated with non-local activity in hippocampus, as I think Brad will have just talked about. This gives the organism flexibility to, say, plan new paths when circumstances or goals change. So for instance, if I'm trained hungry, I want to get to the cheese, but tested thirsty, so I have to go somewhere else, I can figure out how to do that. On the other hand, the classic account of dopamine and temporal difference learning is basically model-free. It works by storing already computed values for different actions. So for the long run, that's computationally less demanding, but it means that it will make mistakes if you test it in different circumstances than it was trained. It chooses the wrong action. So the trade-off between these two modes of sort of flexible versus inflexible choice in computation has been a rich area for us. It's helped us to understand, I think, issues like habits and compulsion and automaticity. However, to make this all work better, we'd like to find kind of in-between strategies that strike some better balance between computation on the one hand and reuse on the other, and in particular, sort of make planning work better. So in particular, many researchers have looked at what's called temporal abstraction, like representing your model of the dynamics of the task in a way that aggregates over multiple steps. This would allow you to simulate the long-run value of candidate actions using fewer steps of computation. The problem with this, actually, both for computer scientists and for biologists, is that, as you can actually see in the subjective function, because I need to choose actions at every step, little step in the maze, right, like each left or right branch point, the longer run trajectory following any particular action actually isn't stable. It depends on all the subsequent choices too. So the future path if I go left at the first step depends what I do later, which depends on where I'm trying to go and my goals are. So this means that the long run model actually isn't easily reusable across tasks, even though it seems like that would be useful to do. Or that's what we thought. It turns out there actually is a surprisingly good approximation of this problem, which actually has been used more in control theory or control engineering than in artificial intelligence. So it's a simple linear equation. Turns out you can approximate this long-run value, V star of a situation or action, by a single matrix multiplication. So it's a matrix M times some vector x r, where r gives you the sort of goals or rewards that you're trying to find, how each location is rewarding. And this is like a single step of a neural network, right? It's a matrix times a vector. So what's M? M sort of briefly encodes this long-run dynamics, right? It encodes multiple future steps of where you're likely to end up following a particular location. But it does so under a default set of assumptions about what actions you're going to take at each subsequent step, like choosing randomly. And it turns out that because the exponentials in this equation, it's able to sort of jointly optimize all the actions and compute V star for whatever it is you should do for some particular rewards all at once by sort of relaxing away from that default policy. So in short, if we can learn this long-run map M or compute it, then we can reuse it over and over again to plan new paths, even if the rewards change. So just to give you some quick examples of how that works, so the first and most direct example is what I just talked about, right? So first in psychology, we often study how animals or people are able to use the representations of task maps to plan new actions by changing the rewards using devaluations or latent learning tasks. So like the hungry to thirsty example I gave. So here's an example where there's a T maze where both sides were initially rewarded and the simulated agent goes both ways at first, but then one of the sides is shocked. And the idea is the animal has to replan a new policy that only goes right, avoids the shock zone. And linear can do this directly, as you can just see from the equation, because all you have to do is substitute the new bad state in the R vector, and M times R gives you, or M times X bar gives you the correct paths automatically, as you see here. So this class of replanning problems is solved easily and kind of more easily than previous as sort of model-based, multi-step-by-step optimizations. But it's more than that, because as I mentioned, this linear RL is approximate, right? But we actually understand the details of this approximation, where it fails, or what biases it has very well. So we can predict particular errors due to approximation that should make in certain tasks that we can contrive. And we expect that to show up as sort of soft biases or increases at the rate of particular types of errors that we can predict that people make, which roots in the use of this default policy to plan actions over multiple steps. And we can actually observe these experimentally in people. I don't have to ever talk about it, but it's reported in this Menajad paper. Let me give you another example. So a more difficult class of sort of flexibility problems arises when instead of rewards changing, the map changes. So for instance, in detour tasks, an animal learns a map with different paths to the goal, including the shortest one. And then in a sort of transfer test, the shortest path is blocked and the animal has to decide where to go, right, and find the next shortest path. So this is challenging for this class of models because it actually changes M, right, the thing that we've sort of pre-computed. So for instance, being on one side of the barrier now it doesn't predict soon being on the other side of the barrier. That's a difference in the long-run dynamics in M. And so we need to do sort of additional computation to fix them up. But one of PyM's insights is what I didn't mention, the initial computation of M is actually a big matrix inversion. And so updating M is actually can be done by sort of standard matrix inversion identities. And it turns out that this case corresponds to a very low rank correction. We're used to add a sort of extra delta M to M to produce the new matrix that includes this new single barrier, right? It's like a rank 1 matrix. And so with this easy step, as you can see, we can actually reuse most of the computation in M and replan and find the next best path again. The other thing that's neat about this idea is that it suggests more generally that our map M for a new environment can be built up computationally by the sum of a set of basis maps like delta M, or the weighted sum of them. And they're tied to features like barriers that the animals can observe. And so the idea is that these pieces could themselves be kind of reused across tasks, and then the way you'll be able to learn an environment sort of is by recognizing them and adding them to your map, right? And so we think that this sort of computational strategy will help us learn new environments faster, and we're working on that. This idea also leads to the last point I have time to mention today, which is sort of what this all has to do with neuroscience. So we think these basis functions for the long-run dynamics map M explain response in the entorhinal cortex. So it turns out there's been a sort of long-standing suggestion, actually, that grid cells in the entorhinal cortex sort of correspond to a basis for M. So the eigenvectors of M, or long-run maps like M, have this sort of spatially periodic with low-frequency components, just like grid cells. What was confusing under the previous accounts, which our new account explains, is why these grid cells in the brain are so stable no matter where the animal's trying to get or which paths he tends to run, which has been demonstrated a lot. So this stability is actually not predicted under previous theories because when the long-run dynamics change, when the goals change, long-run dynamics also change, and so the expectation is that M should also change and grids also change, and they don't. Linear RL actually explains this, the new theory, because it reuses the same map, which was learned under the default policy to optimize trajectories for different goals. This idea about low-rank updates like delta M also has a novel correlate in entorhinal cortex. The correction matrices like delta M are actually produce sort of fields that are yoked to the barrier, and they actually look like a second sort of less famous class of entorhinal cells called boundary cells. So we think what this is leading towards is a sort of common explanation for seemingly different classes of entorhinal responses, all of them understood as a set of basis functions for building up these maps. Okay, so that's it. So in short, we suggest a new way of representing maps for planning that makes it easier to reuse sort of large chunks of computational work across tasks and still replan flexibly when things change. On the theme of this symposium, maybe this is something we as neuroscientists can give back to AI. In general, as I mentioned, these problems of autonomously managing one's own computations have come up more consistently in neuroscience than in AI. And some of our ideas here, in particular, this idea that we can update M with a linear delta M and build it up from basis functions to update maps and learn them, I think is novel both in computer science and in biology. This model also has more implications in psychology that I didn't manage to talk about, especially the questions involving cognitive control and how you can sort of effortlessly suppress habits or reflexes or compulsions and what that sort of costs you. So feel free to check out the preprint, and again, thanks to Hwang. Okay, so now I'll turn things over to our leader, Grace Hwang of Johns Hopkins, for the interactive portion of the panel. So feel free to check out the preprint for the again. Okay, so now I'll turn things over to our leader, Grace Hwang of Johns Hopkins, for the interactive portion of the panel. Our leader, Grace Hwang.
Good afternoon. Let's give a round of applause to our presenters. Great work, everyone. And I also want to convey my condolences to Kanika Rajan and her family for the untimely tragedy. I'd like to start off this Q&A with some speakers' questions that are directly addressed to our individual presenters. And presenters will be able to chime in on other presenters' answers. So why don't we start in order with Conrad first. Conrad. With complex neural systems, there will always be many confounders. Is it realistic to expect studies of population dynamics to control for all of these confounders? If so, how do you suggest?
Okay, so to be clear, my answer is no to that. The long answer to that is, indeed, there's so many confounders. Every dimension is in principle a confounder. And therefore, if you want the drive towards producing a causal model, a model in terms of information transmission, a model in terms of how brain areas affect other brain areas, is probably impossible. And that's not necessarily a problem. No, like, it just means that if we just have observational data, we cannot get there. It is possible if we can perturb, and there's a lot of great techniques. Now we have optogenetics, now we have TMS, we have TDCS. With those techniques, we can get at that. But based on purely observational data, I believe that this is impossible. Now, in small systems, maybe if you give me a slice where I can get rid of the confounders, I can get there. But like for the things that most people in this audience are interested in, that's probably not doable.
Zach, would you care to comment?
Yeah, Conrad's smiling because he knew I would have some chiming in about. I mean, this is the nature of science, right? So you make a hypothesis, you see whether a bunch of variables are explained by the model that you propose, and so you're going to have to make some simplifying assumptions. And under those simplifying assumptions, you can potentially derive causal models. They are simplifying assumptions; they can be falsified. And so that's what you have to look for. Perturbation is the gold standard, but there are good ways of getting at least a decent first guess at things, and we'll see what happens, upon further refinement.
But so far, we don't know if this is true. No, like at some level, it's possible that if we start with a hypothesis and we test simple models, they approximate reality. It's possible that they're not. And so at the moment, we don't have much evidence how good the models are that we fit at actually predicting causal effects. It's very rare that we have both, that we have an interesting model that we fit and that we then test it perturbatively.
All right, let me move on to the second question for you, Conrad. Is mesoscopic modeling justified by the fact that neural computations seem to be very robust against noise in neuronal level lesions for many tasks?
Yeah, this is a fantastic question. Now, like, it is absolutely true that if I take a brain, I take out a neuron, probably nothing happens. It's also true that there's noise everywhere and it has relatively limited effects. And yet, kind of, the question is, does that mean that we can then go from a model that is about the brain in terms of its whole high-dimensional glory to a model that has far fewer dimensions? And I don't think this is a very good argument. And the reason for that is the following. If I take a neural network that I train on something, I can also take out any one neuron. Nothing happens to it. It's just that every neuron does something different. So the big question that we, in a way, have to face with it in the debate that we're currently having is, to which level can we understand brain computation in low-dimensional spaces, or to which level are they in high-dimensional spaces? The fact that they're fault-tolerant doesn't suggest that computation is necessarily low-dimensional. And so in that sense, no, I don't think we can take that as a justification for the possibility of mesoscale modeling.
Why does it not imply that, Conrad? I didn't understand the argument there. I mean, if you have a redundant code, it means that you have a lower dimensional structure there.
So we could, it's about the existence of information, and it absolutely shows that the information that we have in a given neuron isn't essential for the overall calculation. That doesn't mean that every channel codes the same thing. It just means that the signal of interest is contained in many channels. That in no way means that any of the channels are important. Now, like, let's take an example. Let's take vision. I see how far the screen is from me. I can use texture for it, and I can use maybe disparity for that. Those two are two different channels. Both of them allow me to do that. It would be perfectly redundant. I would still know where the screen is. But it doesn't allow me to say that I can basically dump everything together and have only one channel. No, I have a disparity channel and I have a texture channel, and they both allow me to estimate distance.
All right, Conrad, I'd like to move on to the next presenter. Thank you very much. And we'll come back to some of these points when we get to the group discussion. I think there's more to be said. Joseph, we have a question for you. Do your models break in interesting ways? Are there specific kinds of tasks, e.g. moving around corners, abandoning previous reward locations, that are more or less successful?
I'd like to thank Brad for this interesting question about, so this is my neural swarming model that he's referring to. And yeah, so the model does break in certain ways, and what are the most important parametric dimensions along which it breaks? That would have to do mostly with the spatial scales at which both the kind of intrinsic or recurrent swarming dynamics occur, and the scale at which the reward approach dynamics occur. So there's a trade-off there, and those are both kind of trade-offs with the relative scale of the environment relative to the number of, or density of the swarm. And then the other dimension would be the rate of learning. So there's learning rates on both the swarming and the recurrent and the reward approach. And so you can trade off like between kind of random exploration versus direct exploration based on whether you're dominated by the recurrent learning or the feed-forward reward learning. And so those kind of, if you go to extremes along those dimensions and trade-offs, you do get stuck in certain types of dynamics, or maybe these where you get directed exploration or the swarm will just zip to local rewards and this basically stay there. And we did implement a feature where rewards could be captured or memorized, so the value goes to zero once an agent has gone to a reward. And so that's kind of a short-term memory, which enables kind of more long-term foraging behaviors. And there are right, so the swarming dynamics kind of give rise, the recurrent dynamics give rise to kind of a diffusion, almost a gaseous diffusion across the environment that helps you get around corners and get, to find your way through like a hairpin maze or more complex environments. And so that's part of that spatial scale trade-off of like how much pressure there is on that side. So what I showed is kind of like a middle road, parametrically speaking. Hopefully that answers the question.
Thank you, Joe. I'd like to now move on to a question for Brad pertaining to your reverse replay. Does the reverse activity serve as Ariadne's thread enabling Theseus to back out of the labyrinth?
Yeah, I think that's exactly how we're interpreting this, but it's really important to note that we don't at this point have any data to directly support that. This is all correlative. To really show that, we need to directly, remove or force these reverse sequences from happening. But yeah, we think that's exactly right. So these reverse sequences that happen during behavior, we believe, are a way for the hippocampal network to encode the reverse of what is actually happening, to support the idea that at some point later on down the road when the rat is paused, the cells that are encoding that reverse sequence during behavior, those are the cells that participate in the reverse replay, where the hippocampus thinks about the entire path it just took in the reverse order. So yeah, we think this is a way to retrospectively evaluate the sources of some current outcome. Thank you for the question.
Thank you. I'd like to now move to Zach's presentation. Question here is, how interpretable is the message passing algorithm? Would a smart human be able to understand how the inferred network is implementing computation via nonlinear dynamics, especially when it's not a Bayesian inference?
It's a great question. Thank you, Memming. It depends on the particular structure. I think understanding is kind of a funny word. It's like storytelling. We can find properties that are useful for that. And by formulating things as message passing, you can actually structure your knowledge in a useful way. It doesn't necessarily mean that you understand every little nuance of things. And that way there would be some things for us to learn. But you can find, for example, in a graphical model with loops, you find that there's an echo chamber effect where you have some information that could reverberate around if you're just using naive algorithms, but you might have a memory of what messages were sent out that you could then use to discount your future signals coming back in. And that type of thing could be potentially interpretable by a human. But in order to discern that, you'd actually have to look at the content, the actual nonlinear message passing to tell a story about that. So in general, it doesn't automatically give you an interpretation, but it does reduce the space of things that you have to explain.
Thank you, Zach. And I'd like to move on to Nathaniel. You have one question. How valid is the Markovian assumption that underlies most of the RL models when we try to understand behavior?
Well, it's not at all valid if by what you mean is that the input to these models should be the animal's kind of raw sensory environment. Clearly, most tasks are not Markovian in that space. On the other hand, I think that really, whether using an RL model or just having to solve the problem the animal has to solve, you have to map some representation of sort of the current and past events to choose actions to figure out what to do to predict. And the question is, what is the representation that you are using for that decision making? And of course, it has to have, usually in most tasks, history in it. So the Markov assumption just specifies in a formal way what you think the animal needs to have to solve a problem. And then the question becomes, how does the animal brain construct a representation, not just of the sensory world, but of a memory as well, and what parts of memories of what can it use to make this principle hold true? And for that, I think it's perfectly valid. It's a research question. Part of the learning problem is how do you establish representation that does that? But I think you sort of have to, and you can't escape it. I don't think it's like a specific problem with RL techniques. To the contrary, I think it's something that RL techniques expose very clearly as a piece of the learning problem. Thanks.
Thank you. At this stage, I'd like to transition to the more formal group discussion. And I'm going to start off with a question from, I'm going to start with Zach, and then I'm going to ask Joe and Conrad to chime in, and everyone else is welcome to chime in thereafter. So here it is. What do you consider to be the biggest theoretical gaps in understanding the brain that are precluding AI ML from achieving human-level intelligence? And to fill these gaps, is more big data needed?
That's an amazing question. And if we knew the answer to that, everybody would be happy. Theory is really happy about the presence of all this big data, but it means that we need a lot more creativity, I think. There are a lot of neat tools that are coming around and new languages for expressing knowledge. I mean, if you ask me, I think to get a good understanding of intelligence, you need statistics, flexibility, and some kind of compositionality or symbolic computation. And I think that the brain does all these things. But in order to figure out how the brain does these things, we need to have massive scale recordings during complex tasks. And I think that neuroscience has made great strides by using simple tasks, but that time has passed. And there are many ways of solving one of those tasks. So if you really want to challenge the brain to see what it's doing, you have to give it a task where you take away the sensory evidence and reveal the internal model. And for that, I think a moderate amount of complexity is really important. Now, this also gets to the question of that we were talking about before in terms of redundancy and like what is the dimensionality of the actual dynamics that matter. And if you have interesting nonlinear computations that are happening through those dynamics, the more data you have, the more refined you can make your assessment of how the little nonlinear bends in your functions matter. And that's especially important as we move to higher dimensions. But we're always going to need some assumptions under there because there's no way we'll ever get enough data to constrain the dynamics of a very high dimensional system.
Thank you, Zach. Joe? Or Conrad?
Hi, I was just unmuting my mic. Yeah, so this is just to follow on from what Zach said, a transition of sorts to naturalistic stimuli of the very complex behaviors and sensory landscapes of animals experience is something that's going to be really important. And it raises the question of sample complexity, which is perhaps one of the biggest gaps between current kind of machine learning approaches and what we kind of understand about brains. Animals do not see as many examples as modern deep learning networks do. They can learn and generalize very quickly from a small number of examples. So how do they do that? Well, one major difference is that you have dense activation across those forward and backward passes and kind of in these deep artificial or deep hierarchical networks in machine learning, but you don't really see that kind of dense activation in much of cortex in the brain. So you've got kind of sparsity, and maybe sparsity is a fundamental kind of enabler of both hierarchical representations in the brain and the ability for, as Zach said, the compositionality of different hierarchical layers to quickly transfer learning from one domain to another domain. So sample complexity and how you can solve the problem of generalization through changes, through understanding the sparsity of connectivity and how that connects to the sparsity of dynamics and neural activity on top of that structure. So that's kind of a major gap. And that also ties in with the idea that I mentioned briefly in my talk of time dependence. So there's been various attempts, such as recurrent neural networks and LSTMs, of getting time varying learning and processing within artificial networks. But there's tremendous amount of complexity of temporal dependence in the brain, including oscillations and synchrony and phase coding in certain parts of the brain, like the hippocampus and other areas. So there's really a lot of different mechanisms which are yet to be explored in the artificial domain, which might enable flexibility and reconfiguration of basic computational units.
Great, thank you. And Conrad, would you like to add to this collection of gaps we've identified, first with sparsity and then with time dependence?
Yeah, I must say, maybe the main problem is that we're trying to achieve the wrong thing. So today's focus of much of computational neuroscience is to get at mechanism, to get at like little chains of causality. And as I argued, it's very hard to get at causality, but also that's not really what machine learning wants from us at the moment. So if I build a deep learning system, I put a lot of effort into constructing good cost functions. Figuring out what the cost functions are in the nervous system would be super important for that field. I worry a lot about, say, building the right architectures, and people worry a lot about those. And in those, we worry about optimizers. And in a way, this disconnected the questions that seem very attractive from a machine learning perspective, that these connections don't drive so much research at the moment. I think that that's really sad. And so I think if you ask me what's the main missing point here, it is that there's this whole area at the intersection between machine learning and neuroscience that can be opened, that doesn't have the mechanism / causality flavor that neuroscience is usually striving for.
Anyone else on the panel want to chime in?
You're pointing to normative models, I gather?
Not necessarily normative models. I'm just pointing towards models that are organized in terms of cost functions, optimizers, learning rules, and architectures. And there are some that are normative, where you can say you have a cost function that mimics someone's ecological niche. And there's some that are not normative, where it's like, here's a function that's good to optimize. There's everything in between, no? Now, there's for example, inverse reinforcement.
That sounds normative.
To me, normative means two things. It means that there's something that's optimized, and it also means that the thing that is optimized is matched to the problem that is to be solved in the ecological niche of someone. So in that sense, like, I use a slightly different definition of normative. But
right.
Yes,
I've been using rational for that. I've been using the term rational to mean you're optimizing maybe the wrong thing, but it makes sense that you're optimizing that thing because you have the wrong assumptions.
I could come around to rational as the name for this class of models. So different communities use it.
So I just want to remind the audience that it is 1 o'clock now. We will continue our discussion, but I just want to make people aware that it is 1 o'clock. And
bye, everybody.
And so let's please continue with this actually rather important discussion, because it sounds like, you know, there's this disconnect between the presenters and their perspectives based on the answers that Joe and Zach just presented and the rebuttal that Conrad just provided. So I was wondering if anyone wants to add another opinion or to help us come to consensus today. We can be bold and try to it would be awesome to come to some consensus on the gaps in approach that we recommend. I see Nathaniel has his hands up. Go ahead.
I mean, I think one thing that I heard from everybody, which I think everybody agrees, is that we don't just need more big data, you know, full stop, right? We need the right data for the right questions, and we're discussing what those are. I think there's this really kind of naive idea that no one here has expressed, but I feel like I myself once believed long ago, and I think lots of people who haven't thought about this hard believe, which is that we're just going to, you know, measure a bunch of stuff about the brain and figure out how it works and go tell the professionals in engineering how to solve their problem. And, you know, the professionals in engineering are smart people who are professionals who know a lot of stuff. And if, you know, big data is going to be a piece of helping them from neuroscience solve their problems, it's going to involve us talking to them about what they need and them talking to us about, you know, how we can interpret our data. And I think there's lots of opportunities there. And I think, you know, big data and the right tasks is an important piece of that. I think figuring out where the problems actually are in AI, if that's what you're trying to solve, and asking those people, as Conrad said, is another thing. So I think in that respect, I think that everyone's right.
Great.
Mike Chapman had a question that actually relates to this. He said, shouldn't the search for hypotheses be guided by structure and function, ontogeny, phylogeny, and other biology things? And I think the answer is absolutely. And that's, you know, there's some like legacy code that we get, that we inherit, that's not necessarily the best of all possible legacies, but it's what we manage to get from here to there. And Paul Chisek, I think that's how you say his name, but I don't know, has an interesting theory about how the different parts of the brain are structured according to actual ecological problems that happened along the way of evolution. So I think that that's an interesting architectural set of constraints connecting to what Conrad had said as well.
So that sounds like yet another important gap to understand the relationship between structure and function. Great. With that, I think I'd like to, if there's no other hands, I'd like to move to another panel question, and this one will be directed first at Brad, and then anyone else feel free to join in. How can we translate decades of spatial navigation studies into spatial, sensory, and motivational signals to explain foraging under uncertainty in the context of machine learning?
Yeah, great. Thanks for that question. Yeah, so a lot of work on rodents and more recent work on humans has suggested that during periods of purposeful memory retrieval, the hippocampus is really good at generating sort of predictive sequences. So in the rodent, for example, in an open field where the rodent has to navigate to a learned location, what we observe is during the periods of sort of deliberation before the animal actually takes an action, there are brief sequences that encode the spatial trajectory starting from the animal and ending at that goal location. That presumably requires some knowledge about the environment. It presumably requires some level of learning. We have not yet quantified the extent of that, but presumably that is required for an animal to engage in sort of purposeful memory use, to retrieve the previously stored information in a meaningful and present it in a meaningful way. During random foraging, though, which I think is part of that question, we don't see this. We don't see this sort of, we still see sequences that are expressed, but they don't seem to correlate with the animal's actual behavior. They seem to be completely independent, suggesting that there might be two distinct mechanisms. One in the hippocampus, which is just generating sequences of information that might or might not lead to important locations or information content. And another region, maybe the cortex, that's listening to that and evaluating the content or the salience of those paths to decide whether or not it should act upon that information. Right. So that would be my answer to that. That's what I have gathered from the decades of work looking at sequential reactivation in the hippocampus.
I see Nathaniel has a comment. Go ahead, Nathaniel.
One thing to add to that, which I think this is the history. Can you hear me? I have some network problems. The history of the hippocampus, I think, is a good example of Zach's point that we want big data kind of in the right tasks or complicated tasks. So I think so much of the work on almost all of it on animal representations of space in the hippocampus and replay and preplay and all of it occurs in the context of sort of minimal behavioral task for the animal, right? It's basically random foraging. Brad's study is one notable variant, actually. And I think it's because the place fields were so exciting that people became sort of part of sensory neuroscience. Like the question was, how do these neurons get their response, basically? And that sort of dominated the field for decades. And I think this is a great example where now it's time to sort of put them back and ask, how do animals use this stuff to actually behave? And I think that's a very exciting question, which again, I think is sort of in the spirit of the sort of thing that Zach was mentioning, for instance.
And would you have some thoughts about new experiments of design to run experiments with rodents in complex environments to get at these questions?
Yeah, I think, to some extent, the kinds of experiments that, for instance, Joe was modeling, foraging for rewards in dynamic environments and studying the choices and the neurorepresentations and how experience drives neural activity and how neural activity plays into subsequent choices. I think that's going to be where the action is, and it's very exciting.
I'd like to ask you a question as a follow-up to that. One of the things that I find the most challenging is how to think about learning in these contexts, because you only get one shot to see the first shot at something, right? So learning, the process of learning, you just never get a lot of data about any one thing that you've learned. How do you think about good tasks or good experiments to design a learning experiment that would generalize?
Yeah, I think to get at that question, you need to be able to monitor the same population of neurons across long periods of time, across many different days or repetitions of a novel task, so that you can monitor network activity as the animal transitions from that task being novel to that task being familiar. So I think long-term recordings are going to become more important for addressing that point.
So if that would be really helpful if you have something that the animal learns slowly. But there are many things that an animal learns quickly, like they see something for the first time, and then recording over time is very difficult to, does it matter?
Yeah, that's a fair point as well. Yeah, there are many examples of one-trial learning, especially in the rodent literature. Right. For those tasks, I imagine that it'll be important to just record from a sufficiently large number of rats so that you can identify in each rat the neurons that are uniquely encoding that experience, so you can see how their patterns of activity or potentially their connections or functional connections between other neurons that also encode similar information, how those might change across that single experience.
Yeah, I wanted to follow up on Nathaniel's point that we kind of need data for
a really broad set of prompts. Now, like it's not enough that it's just rich. So when it comes to one of the prompts that are really hard for AI at the moment, it's multitask learning, where you learn one thing, then you learn the next thing, and then you learn the third thing. And for us humans, it's generally the case that, like, hey, we kind of learn stuff about the domain. We keep getting better. If we do that with our algorithms, they tend to get worse, and then we use all kinds of hacks to avoid them from getting worse. And yet for the nervous system, it seems to be very natural that it keeps getting better. And I think basically, therefore, we are so focused in neuroscience of giving one experiment for a long period of time and maybe varying a very small thing. Basically, how animals deal with learning different things one after the other is one of the big questions that we need to look much more after.
Absolutely. Joe, you had your hand up?
Yeah, well, I wanted to follow up on, I guess, the two previous points. So I had a study with Jim Knaram's lab, who does more random foraging experiments in rats, where I was able to establish a behavioral relationship with these investigatory behaviors where the rats will laterally scan their heads back and forth. And I was able to show that persistent abrupt changes in place field map could be associated directly with singular behavioral events. So the animal would stop from running and just look out into the environment, and then on the very next lap through one of these circular closed loop tracks, a place field would appear there, and that would be a stable, persistent place field, which the long history of hippocampal studies have suggested may be a neural basis of episodic memory. So that's kind of like the canonical example of one-shot learning in mammals. So there's possibly a very strong interplay between volitional actions and behaviors with respect to the environment and how one-shot learning is implemented in hippocampus and perhaps other parts of the brain. And then this also ties into the idea of sparsity versus density. So that these hippocampal memory networks are sparse, with maybe 2 to 5% of possible connections actually existing. And so how does that a structural sparsity enable the learning of these kind of very high-dimensional, high-order events across an animal's lifetime in a way that does not give rise to catastrophic forgetting that you might see in these dense artificial neural networks. So maybe sparsity and kind of these pre-configured networks indicated by the pre-play phenomenon that have been revealed kind of show that maybe this is underlying kind of dark pool repertoire of activity which can be brought forth by fairly simple but transient and powerful learning episodes.
Thank you, Joe. I think that was a great summary of ideas. I'd like to shift gears and take one audience question at this time. This has to do more with optimizer functions. So what is the optimization function, which it seems to me that a lot of optimization approaches are defined by the observer. In that sense, it is a subjective phenotype. How can we introduce evolution into optimization? This question is from Dr. Fidel Santamaria.
Well, there are multiple ways now how evolution plays a role. So the first one is that if I try to understand a new animal that I might not know so much about, I will look at the ecological niche. I assume that evolution has allowed the animal to be good at the things that the animal is doing, maybe like forage efficiently, maybe solve those prompts. And then in a way, evolution gives me a justification about why an optimization function should have a certain role. But there's a very different way how we can think about it, where you can say there exists the need to optimize different things in an animal's life and in the way the animal interacts with its environment. And maybe, therefore, there's different optimizers in different parts of the brain. And evolution has kind of given us this collection of optimizers somehow jointly. And in that sense, we might rather ask for like which were the optimization problems that were important in our far past. So it's a very rich question.
Indeed, I see Zach has his hands up.
Yeah, it's more a follow-up question to that. So if you think about multiple optimizers, can't you just put all of that together into one big optimization that's context dependent and therefore depends on more of the state of the world?
In principle,
the original
yes.
question again, right? What's that?
Yeah, in principle, yes. Now, but the question is if this is a good way of thinking about it. So if we have the strict optimization idea where you have one cost function that you optimize, then it robs you of certain kinds of flexibility, and it might be computationally much harder. Now, you can say that if I optimize a given thing at a given point of time, now, like right now, I'm not maybe optimizing my food intake, and I might dedicate absolutely no cognitive resources to optimizing my food intake. And similarly, if you're like a baby, if you're at a different developmental stage, you might want to run a very different cost function that you optimize. So this idea that there's one global cost function makes things computational harder. It also makes it easier maybe to game you. If I know that you have one global optimization function, then it might be easier for me to predict you. So there can be other reasons why you wouldn't want to follow a strict optimization framework.
So
Anyone else?
If you're going to break an optimization into separate optimizers, then you are imposing some kind of modularity, right? And so this gives you a hierarchical structure. And then you get involved in what am I going to plan? Like, do I have long-term planning over which optimization am I going to run? And then you have the local optimization, like now that I've conditioned on that. And I think that there's good evidence that we do conditioned perception and we do conditioned action. Like we're not constantly planning everything. We have some, but I think a lot of that can be subsumed under resource constraints. We're not able to optimize. We're only able to locally optimize. And then the question of what is the optimizer kind of boils down to what are the constraints?
Yeah, I mean, we're fully in agreement here.
Well, we can't have that.
Sorry about that. Let me just say that I'm getting hungry.
Uh-oh.
Now you brought it up.
Sorry about that.
I guess at this point, there's sort of one point that we hadn't had a chance to talk about today, which has to do with, comes from Conrad's presentation, in which he describes the need to be able to measure information flow in the brain. And I've not heard much of that discussion during the panel, and I think we're probably going to have to wrap up soon. So I wanted to open the floor to see if people think that's an important theoretical gap that we need to address in addition to all the things that we've talked about today.
But to be clear, I mean, my point was that it's kind of impossible. So the question is, do people disagree with it being mostly impossible? And do people think that it would really advance us if we could get there?
Well, my whole presentation was predicated on the idea that there is information flow, and information comes in over time. So I think, yes, there is, it gets to some parts of your brain first. It gets to some ideas first, and it takes time to make connections. Maybe you could even invoke thinking fast and thinking slow. Under some conditions, it's going to be, I agree, impossible with the amount of data that we have to discern some of the fine details of that information flow. But in other cases, I think it's pretty clear that you can find the information flow. And some of the early work in very fast perception, you can measure just synaptic delays of how long it takes to get there. And while that's not a knockout for any model at all, under what we think of as reasonable models for like how long a synaptic delay takes and whether you would have sort of retroactive, like backwards signals propagating with faster delays than other backwards signals. I mean, it would take a lot of weird modeling to be consistent with the forward flow of information that we observe in terms of experiments and still say, oh, well, there's a confounder in time. So I think information flow can be very well defined, and I think it's a useful concept that we should push on.
I agree. It can be defined, and it's a useful concept. The question is under which circumstances we can practically measure it.
I think Joe had a comment before we go on to your next question. Joe, did you want to comment?
Basically, probably what Conrad was about to say, which is that, so are we talking about a distinction where, yeah, there is this information flow over recurrent networks that's very difficult to interpret, externally, but maybe that's the critical computational basis for the animal to create relevant information that's meaningful as far as its future actions and cognition, versus our ability to observe the brain of another animal as an external party and interpret that within our own experimental information context. So you could have these critical types of information flow that Zach talked about, but then also but have this kind of maybe intractable or very difficult problem of measuring it and understanding what our perturbations are doing to the complex information flow within these systems.
Conrad?
Well, I think we're all in agreement here.
Wow, this is fantastic. Would one of you care to summarize and prioritize the 4 main gaps to bridge machine learning and theoretical neuroscience and experimental work?
More ideas. We need more ideas and more data.
And we need to rethink the old ideas.
Do we need to worry about the mismatch in dimensionality between ML systems and the brain? I believe there's still, I think, disagreement between the 3 theorists who presented where
Yeah, I agree that it's not clear that there's really a fundamental mismatch. If anything, the brain is they're both big dimensional, right? The question is can you understand it in terms of simpler terms? You might mechanistically need a lot of units in order to give yourself the fertile soil to give the computational dynamics that you want. That's sort of the way that I think about it. If you had an arbitrary neuron with multi-dimensions, et cetera, then you might not need so many neurons. But when you have constraints on the biology and constraints on the functions, then having a lot of neurons is just a way of enacting the right kind of nonlinear computational dynamics in some lower dimensional space. But you also have to get to that lower dimensional space by selecting the right features. And so the input is high dimensional. So I think that there are roles for both of those kinds of computations.
And the question is where we need low dimensionality. We might assume low dimensionality in the processing, and I guess I'm just more pessimist than Zach there. Or alternatively, you might assume low dimensionality on the side of learning rules and architectures, and that might be in fact a place where a lot of us might be able to meet.
Yeah, I would certainly agree that you get inductive biases from both of those, architectures and plasticity rules. Absolutely.
Well, great. I think we're well over time. So if anyone has anything they want to say, give me your hand. Otherwise, Zach has something he wants to say.
I want to say thank you, Grace, for organizing this really fun panel discussion. And thanks everybody
thanks very much
for listening in and for
Thank
asking
you, everybody. We'll see you guys soon virtually. Take care.